Good morning or good afternoon. Hey guys, and welcome to another great week. The last week before spring break. Woo! And thank goodness we're having one. Yes, good Lord, we need one. Okay, guys, so welcome to today's lesson. It will be on classification. So far in evolution, we've talked about how evolution works through natural selection. We've talked about how evolution is able to create speciation, which is the creation of new species from old ones. We've talked about how all animals and all organisms are related in some kind of way through their DNA. We looked at DNA profiles of different organisms to see whether or not fish were the most closely related to tetrapods. We've looked at a lot of really cool things. And today we're going to look at one of the coolest, which is how we take everything we know about evolutionary relationships in organisms and we plot it onto a giant family tree. We call it the tree of life. It's the thing that shows us that all organisms are related to each other, and we have a lot more in common than we do differences apart. And this is a really cool study of classification. It might seem like the most boring thing in the world, but actually, it was the study of classification that led Darwin and Wallace to their theories of natural selection, because they noticed that organisms were more closely some organisms were more closely related to each other in shape and in look and in environment, and that gave them the idea that, hey, maybe the environment helps to select for a phenotype that allows them to survive. So classification is a very old study. It actually predates everything that we knew about evolutionary biology, but the great thing about evolutionary biology and about evolution by natural selection is that, like any good hypothesis, it allows us to make predictions and to test those predictions against what we found in nature to see if they're true. And what we discovered in evolution helped us, was not only supported by taxonomy, but actually helped to clarify and correct mistakes that had actually been made previously by taxonomists who didn't know anything about evolution. So taxonomy and evolution have been working together ever since natural selection was discovered, and today the two are inseparable. So let's go ahead and take a look at classification. What does it mean? Okay, so the first thing is that classification is basically how we group organisms based on similarities. Now, those similarities could have a wide range. They could either be molecular, like DNA similarities, or they could be anatomical, and we'll take a look at that in a second. But basically, just looking at organisms and trying to see, are they related? And if so, what are they related based on? Okay, and then we have taxonomy. Taxonomy is the science of naming organisms. And to make things easier on scientists, classification tries to name closely related organisms with close names so that we can tell that they belong to the same group. So for instance, if your last name, like my last name is Garner, right? Okay, cool. Well, most of my family, at least on my biological parent side, are named Garner, and that allows me to know, hey, I'm closely related to them. And in the same way, organisms that have similar names are often more closely related. We try to put them into groups based on their names. So that's what taxonomy is. Okay, so what is the basis for our modern taxonomy? Well, we like to use homologous structures. So remember, that's when we have the same bone structure but a different function, like a whale's limb versus a cat's paw or whatever. Similar embryonic development. So remember, we talked about the fact that all animals, no matter whether they're a fish or humans or anything in between, they all had similar embryonic development, which suggested a common ancestor. And then similar DNA, RNA, or amino acid sequences. So if you remember when we were talking about, back in the lab day when I showed you guys that blast sequence and everything like that, we were trying to compare DNA sequences and genes to see whether or not organisms were closely related. And that is one of the big ways, and again, basically the most important and accurate way of showing whether organisms are similar. How I like to think of it is your genome, your DNA, and the collection of all your DNA is like the book of life for you. It's your book of life. And specifically, it's your species book of life. So all humans have the same book of life, and all with very small modifications. All whales have the same book of life, all plants have the same book of life, everyone's got their own book of life, every species. And if you compare all those books, if you were to sort of open them up and read through page by page, what you notice is that if the organism was very closely related, so say we compared the pages of a chimpanzee's book of life to a human's book of life, their pages would be almost identical. It would only be certain sequences that would be rewritten, like almost like a second version or a second printing of that book. And those differences are actually be in the genes that would code for the phenotypic differences between the species, which is really cool if you think about it. So really what we're doing is we're just holding up everyone's books of lives and we're seeing how they've been modified and changed over time. And of course, the assumption is that way back when there was one book of life that sort of spawned all these books of life, that single common ancestor. And that's really what we mean when we look at similarity in DNA sequences. We're just comparing books of life. I hope that's a good analogy for you and it helps you out there a little bit. Anyway, so classification. So the groups that we split these into are called taxons, and it's a wide category, and different taxa are showing different levels or sizes of organization. And there's a hierarchy of these taxa from the biggest to the smallest. So when we want to start off looking at an organism, maybe we don't know what it is. We want to start with broad ranges of classification. We want to be like, okay, it kind of looks like this group of animals, and then we want to narrow it down from there. And so we have these preset groups that allow us to do that. And those preset groups are going to be domain, kingdom, phylum, class, order, family, genus, and species. So it's going to go from biggest with domain, that includes a ton of organisms, all the way down to species, which only includes one type. And we kind of go down this list when we're trying to name a new species so that we can place it in just the right group so that we know it's close to its evolutionary ancestors. Okay, so our domain is our broadest, and our species is our smallest. So if we were going to go ahead and see if we could take the grizzly bear and determine what classification it would fall into, we would first start off by saying, okay, it's an animal, so it's in kingdom Animalia, and that includes all animals, so we can see, wow, it includes such diverse things as a sea star, a red fox, a grizzly bear, a lot of stuff. And then we would say, okay, so it's part of the phylum Chordata. That's a slightly more specific classification that's talking about everyone having a backbone. Well, that involves the snake, the squirrel, the fox, and all those, but obviously not the sea star. It doesn't have a backbone. Then we go to mammals, which are organisms that have fur and mammary glands and give live birth. And we can see that that includes the panda bear and the fox and the squirrel and all that, but not the snake. And then we go to carnivora, which means that they only eat meat. Okay, so again, here we have another shifting down because the squirrels don't eat meat. And then we have Ursidae, which is including only bears. And so we see that only bears are left. And then Ursus, the specific genus that actually cuts out the panda bears. And then the species. So we can see that this is just an ever increasing specificity of organization in which we're able to just take an organism and put it down to its most specific name. So the grizzly bear is Ursus arctos. And we can see here that this is how we've done it. So of course, this chart doesn't show the domain. The domain would be eukarya, though, and we're going to take a look at that in a second because these are all eukaryotes. They're all multicellular organisms whose cells have a nucleus. But so a good way to remember the various different classifications is through this mnemonic. Dear King Philip came over for good soup. You can come up with your own, but this is the one that I learned and the one that I think everyone learns to be able to remember these orders and remember that it goes from the biggest at domain all the way down to the smallest at species. Okay, so if we were to classify humans, we of course are domain eukarya. Our cells have nuclei. We're eukaryotes. We're in Animalia, Chordata because we have backbones, Mammalia because we have hair and mammary glands. We are in the order of primates, so we've adapted to climb trees. But we're in part of hominidae, which are bipeds, so we're able to walk erect. Genus Homo, so we're able to have a large prefrontal cortex in our brain and use tools. And then of course our species specifically is Homo sapiens. So it's pretty cool. Okay, and you won't have to do this now, but you'll do this in your practice for later. So the first taxonomist arguably was 2,000 years ago with Aristotle. Of course, there may have been many others, but that's the one we have record of. Aristotle divided organisms into two big groups, plants and animals. Woo! Not plants versus zombies, that came later. He then subdivided them by their habitat, whether they were land, air, or sea. So he was like, there are land animals, there are sea animals, and there are, what was the other one? Air animals. I think I got all three. And I guess he thought the same for plants, although I don't know if I've ever seen a flying plant before, but who knows? Anyway, so he did that, and then it was like a long time later that this guy Carolus Linnaeus came along, and we consider him the father of taxonomy. Yep, he's another old white dude who looked at a bunch of plants and animals and was like, I think we should call it this. But anyway, he was the first well, he wasn't the first. He was the first really widespread taxonomist. So he was organizing animals into different groups and plants. And he actually classified organisms by their structure, which was a great idea because, as we know, those homologous features are better for classifying organisms than just their outward appearances. And he developed I know, I'm sorry, the words are going off the screen, but he developed a two-part naming system called binomial nomenclature. And we're going to go ahead and take a look at that. So binomial nomenclature is a two-part naming system, and it includes whatever organisms, genus, and species. And those are its special names. So you can think of it as its first and last name. It's in Latin because Linnaeus sucked. No, it's in Latin because that was the most common language of the day for using this, and it's either italicized or underlined when written. So if we're looking at this little robin over here, that robin's name is Turdus migratorius, which sounds like it's a migrating turd. But no, it's the American robin. And actually, to tell you the truth, looking at this picture, because I am super into ornithology, I believe that is actually the European robin, not the American robin. So whoever put this together, ha-ho, your birding skills are lacking. Anyway, Turdus migratorius, the migrating turd, or the American robin. I mean, heck, how could you forget that name? But really, this just means that when you look at this thing, and if somebody were to ask you, hey, is that an American robin? You could say, yes, it is Turdus migratorius, and they would know you're talking specifically about the American robin because the European robin is not named Turdus migratorius, even though the picture is of a European robin. So that's fun. But basically, what I'm trying to get at here is that a species' taxonomic name, its binomial name, its binomial nomenclature, is specific to it and only to it. And that's really important because there can be a lot of confusion about an organism's name, right? So we have three different organisms here, three different bears here, and they're all bears, right? And so you could be like, oh my gosh, it's a brown bear, that's a white bear. But maybe if you said that's a white bear, people would get confused if they didn't know you were talking about a polar bear and might think, oh, it's a panda bear because pandas are also white. So to get rid of the confusion, we've come up with these special names for them that cannot be confused. I mean, how could you confuse Alioperda melanobluca or whatever that is with Ursus maritimus? I mean, that's impossible. You would have to know what they were talking about so long as you knew what those names referred to. Okay, so binomial nomenclature is a two-part naming system, and we use the genus and species for the organism's scientific name. It's like their first and last name. So it allows us to accurately and uniformly name organisms. It uses the same language for all names, which is supposed to be unifying, although Latin is very hard and a dead language now. And so, we can get special names. They aren't nearly as confusing as common names, which can sometimes be a bit weird, like seahorse, that's obviously not a horse. It also helps us to solve this problem. So way back in the day, when people were looking at organisms, they would have common names for them. So some might call it a mufette or a cinquier or a skunk or a cheche. Every language would have a different name for an animal, and not only that, but even within a language, we would have different names for organisms. And it was very difficult to tell what anyone was talking about because no one knew what animal everyone was referring to. So, for example, when I grew up, over where I lived, there were these big old cats. And those cats, some people called them cougars, some people called them mountain lions, some people erroneously called them bobcats because a bobcat is a whole different kind of cat. And so when someone would say, yep, I saw a big cat up there in the mountain, or I saw a big cougar, you're like, okay, so what are you talking about here? Are we talking about a mountain lion? Are we talking about a bobcat? You're talking about a neighbor's cat that was wandering around and just happens to be huge, right? But if you use the specific scientific name for an organism, it's impossible to confuse. So when everyone came up with Mephitis for the skunk, or whatever the scientific name is for a mountain lion, everybody knew what everyone was talking about. And that allowed everybody to agree and also to sort these organisms based off of their evolutionary history rather than what somebody just decided to call them when they saw it. So hopefully that makes sense. Basically, scientific naming is to allow us to know exactly what we're talking about and to sort it by its evolutionary group. So there are different levels of classification. We have the six kingdom system, which sorts all organisms into six kingdoms: eubacteria and archaebacteria, protista, plantae, fungi, and animalia. And then we have the three domain system, which is sort of, I'll let you know, the three domain system is actually sort of over the six kingdom system. It's like, it's an umbrella. It covers the six kingdom system because bacteria and archaea, excuse me, are both part of prokarya, which are prokaryotes, so they're single-celled organisms without membrane-bound organelles. And then we have eukarya covering protista, plantae, fungi, and animalia, and those all have membrane-bound organelles in the nucleus. Archaea and bacteria, eubacteria don't. So those are the two big prokaryote versus eukaryote we talked about earlier. We just separate prokaryotes into archaea and bacteria to be a bit more specific now. So hopefully that makes sense, right? We just split them based off of their specialized features. Okay, so domain is what I was just talking about, eukarya and prokarya, and prokarya includes eubacteria and archaea. Those are the broadest, most inclusive taxon. There are three. So archaea and eubacteria are both prokaryotes, no nucleus or membrane-bound organelles, and eukarya have a nucleus and membrane-bound organelles. Archaea are the ones, we call them the ancient ones. They're the ones we think that eukaryotes evolved from. Archaea are kind of the in-between step between prokaryotes and eukaryotes, or bacteria and eukaryotes. Archaea are really good at living in super harsh conditions, like conditions that would melt metal. They can live in those, and it's crazy. And they may have been, again, the first cells to have actually evolved. So we can see a picture of them here. They're just sort of blobs, not very interesting. They don't have a nucleus. They don't have membrane-bound organelles, but they can survive these extreme conditions. That's why we call them extremophiles. They love extreme conditions. So they can live in sewage treatment plants, thermal vents. They can live anywhere along the earth. Then we have eubacteria, which are the oldest ones. Actually, it's funny, we call archaeans the ancient ones. Eubacteria are actually the oldest group. And they cause a lot of human diseases. As we all know, we've all encountered bacteria in some ways, especially if you've had strep throat. Oh, God. And they live in basically everywhere on the earth. And so here's a bunch of bacteria. And again, they have no nucleus or membrane-bound organelles. The blue stuff that you see in those little thermal images of those, that is their DNA, but it's not contained in the nucleus. So many bacteria are very important to the environment. Most of them don't do bad things to us, but some of them do. And they can live in the intestines of animals, and that can be a good or a bad thing, depending on whether or not the right ones are living in your intestines. That sounds like a commercial. Are the right bacteria living in your intestines? Stay tuned to find out. Okay, so, and then we have domain eukarya. And eukaryotes, remember, have a nucleus and membrane-bound organelles. They include protista, which I think we looked at under the microscope. Those were those weird little swimmy things that were swimming around. You remember that Leeuwenhoek found and stuff in the little water droplets, if you guys remember looking at those. So those are protists. Those are actually, those are eukaryotes. Fungi, so mushrooms and all that stuff, plants, I mean, you know, and animals. Hopefully you know what those last two are. If you haven't, go outside, but don't get the coronavirus. Okay, so three domains of life. Those are the bacteria, archaea, and eukarya. And those cover every other organism on Earth. Everything falls into one of those three categories. That's how broad a domain is. Oh, and this just tells us about each one of those. I'm actually not going to go into these right now because we're going to explore these a little bit later. So fungi, plantae, they all have different features that help us to know what they are, but we're going to get into that later. Okay, so here are those six kingdoms again. And here's some of their traits. Moving on. So we can see that this places all organisms on the tree of life, and it places them there based off of features that make them very similar. So all plants have similar features, all animals, etc. And this all leads us back to there must have been a universal common ancestor that gave rise to all of these. So all of these different books of life represented by these six kingdoms and three domains, all these books of life are all related, and they share many huge parts of them are going to be almost identical. And it's the parts that are changed that make them different from each other, that make a plant, and an animal, an animal. So the basis again is for this is homologous structures, embryonic development, and DNA and RNA. Going back to what those are, homologous structures, if you remember, are those similar bone structures that we looked at previously. So each organism has the same bones, just been modified in shape and size for different purpose, but we can see they all have a humerus. They all have the two bones connected to the humerus. They all have some kind of wrist bone, and all those bones are related. And then we have all the finger bones, which are all related as well. And each one has just been modified to a different purpose, but it has the same internal structure, suggesting a similar evolutionary ancestor. We also can see similarities in their embryonic development. So we can see, wow, holy cow, the fish embryo looks almost exactly like the human embryo, which looks almost exactly like the pig embryo, et cetera, et cetera. And so this embryonic development, because it is so close in every organism, we know that this is great evidence for the fact that there was probably one common ancestor that had an embryo like this, and then modifications and changes to those embryos over time have created the modifications and changes you can see happening to each of these organisms to make them different from one another when they're fully developed. So homologous features, embryonic development, and DNA comparison are the three characteristics that are the basis for modern taxonomy. And then we get phylogeny, which is the last thing we're going to talk about today. So phylogeny is when we are making basically a map or a family tree of organisms based off of their characteristics. And the phylogenetic tree is going to branch off, and each branch is going to show evolution. So if you look at this here, usually they'll come with a time scale on the side so you can see how long it took for this to happen. Each one of the places where we come to a T intersection, so this right here, they're not marked, but they're known as nodes, and this represents a common ancestor. So we can see here, here's humans, and here are mice. And at one time we shared a common ancestor, and that common ancestor split off and eventually became mice, and one group eventually became humans. Now, were there a lot of intermediate steps between this? Yes. This is the thing about phylogenetic trees is they can be super specific or they can be more general and broad. And this is a far more general or broad phylogenetic tree. It's just showing big differences between organisms and showing that, yes, there was once a common ancestor between platypus and birds or between marsupials and humans, but those common ancestors can go sort of far back. If we were to have if we were to make a phylogenetic tree that included every organism we knew about in every relationship, it would have so many branches, it would look like, I don't even know what it would look, it would just look like a series of tightly packed lines. And so we have to kind of simplify it a little bit to show phylogenetic relationships here. But it allows us to show how closely organisms are based off of their most recent common ancestor. If they shared a more recent common ancestor, they're more related. So the mice are more related to the humans than they are related to the platypus because they share a more close common ancestor with humans than they share with the platypus. This was the last common ancestor between the platypus and the mice. In the same way, the fish last shared a common ancestor here. The fish shared a common ancestor, or excuse me, sorry. The birds shared a common ancestor with the group that would become the monotremes and the marsupials. They shared that here. And so the birds are less related to the marsupials right up here than the monotremes are. The monotremes, because their common ancestors right here, are more closely related to marsupials. So the shorter distance that you have to travel from the most recent common ancestor is going to show how closely related groups are on a phylogenetic tree. Okay, so what is a cladogram? So that was a phylogenetic tree. It's a family tree to show evolutionary relationships between organisms. A cladogram is going to show how organisms are related based on their shared or derived characteristics. So you notice that the phylogenetic tree we just looked at had no characteristics. It just had names of organisms. The cladogram adds names onto this, so based off the development of certain things. So a cladogram is going to look very much like a phylogenetic tree, but instead of just including the names of organisms, it's going to include certain features that they share or that have evolved. So between the salamander and the turtle, organisms evolved eggs that they could lay on the land. And so the salamander only lays its eggs in the water. It came before that evolutionary history. And the turtle came after, so it lays its eggs on land and buries them in the sand. Okay, so anything to the right of one of these evolutionary traits has that trait. Anything to the left does not. So salamander, grouper, lamprey, lancelet, none of them have amniotic eggs, but both the turtle and the wolf do. Okay, and if we go down here to jawbones, okay, the lamprey and the lancelet, they don't have jawbones because they're to the left of the jawbones, but everything to the right does. So the grouper, the salamander, the turtle, and the wolf, they all have jawbones. So this is a way of showing when certain features evolved that help to separate these groups and to show how related they are based off their traits. So here we can see that with the humans and with primates. So whoops, we can see here that the human here, we can see that, let's see here, downward pointing nose or central eye area for more acute vision. So over here, these guys have eyes that are more on the side of their head. These guys have eyes that are more on front. These guys have more downward pointing noses, where these have more upward pointing noses. We see that these guys all have tails. These guys lost tails, right? So we can look at this and see these are the features that make humans characteristic from various different species of primates. It's pretty cool. So this is our cladogram. Okay, so what does a cladogram show? It shows when certain features and traits evolved, and it helps us, based on that, to show which organisms are more closely related, which is pretty cool. So it's and then okay, so then this brings us to our final tool. Okay, so far, just to recap, we have been able to make family trees of organisms, those were called phylogenetic trees, that showed their evolutionary relationships but no traits. And those phylogenetic trees are based off of homologous structures, similar embryonic development, and on their DNA sequences. So how closely related they were based off of their books of life or their DNA. Okay, we then looked at cladograms, which showed very similar information to phylogenetic trees. It just included traits in when they evolved to show how groups are related. And then finally, what we can use is a dichotomous key. So where do dichotomous keys fit in? So let's say that you encounter a new organism. You don't know where to place it on a phylogenetic tree or a cladogram, but we already have these lovely kingdoms and groups that we could sort it into, and we know that each kingdom and group comes with certain features that allow us to identify it as part of that group. So a dichotomous key is a quick tool that biologists use in the fields to be able to look at a new organism and to try to sort it or identify where it should go in this big tree based off of its characteristics. So it's used to identify organisms. You read its characteristics, you kind of create a little chart, and then you see if you can figure out what it might be closely related to. This is used to either place it on an evolutionary tree or to identify it if it's a known organism already. It might already have been named, and you can use this to determine if that is true. So what is a dichotomous key? It's just a list of traits that we can use to determine whether that organism is a certain type of organism or not. So if we were to look at all these tentacled organisms over here, right, we have all these different organisms with these tentacles hanging down, and we were wondering, okay, which organism is what? Okay, what are any of these an octopus? Are any of these a sea anemone? Are any of these a hydra or a jellyfish or something else like that? Then how would we be able to tell? We would use the dichotomous key. So let's pretend you've never seen these organisms before, and you go to this key. Let's see if we can tell who is what. Okay, so let's start with this guy right here. So I'm going to look at this one. I'm going to look at the features it has, and I'm going to say, okay, now I'm going to look at the key. Are tentacles present? Yes. Tentacles are present, and so I'm going to go to 2. All right, if tentacles had not been present, I would have said, okay, tentacles are absent, and I would have gone to 3. But because tentacles are present, I go to 2. 2. Does it have 8 tentacles? Let's see. 1, 2, 3, 4, 5. Wait, 1, 2, 3, 4, 5, 6, 7. I see 7 tentacles, but there's probably an 8th one hiding because I don't have an option to have less than 8 tentacles. So I'm going to go with it has 8 tentacles and it must be an octopus. Okay. Now I'm going to go down to this organism over here. What is this one? Okay, so I go back to the beginning. Are tentacles present? Yes, I go to 2. 8 tentacles? Looks like a lot more than 8. So I'm going to go with more than 8 tentacles. I'm going to go to 3. Do the tentacles hang down or are they upright? So these tentacles, some of them are hanging down, but a lot of them are kind of coming out of the top of the organism. And so I'm going to interpret that as meaning upright, and that is my sea anemone. Okay, so let's go to this one here. Okay, so this one, are tentacles present? Yes, go to 2. All right, does it have 8 tentacles? 1, 2, 3, 4, 5, 6, 7. Okay, so interesting. I don't know, more than it might have more than 8 tentacles, I guess, to go down to 3. Maybe that's my sea anemone. I don't know. I'm doing so well with this one here. This is maybe not the best dichotomous key to use. Let's try this one. I think it's a jellyfish. Okay, so let's try this one. Okay, so tentacles are present. Go to 2. Does it have 8 tentacles? I can't really see, but I think it has more. So more than 8, go to 3. Did the tentacles hang upright? No. Or so no, they hang down. So it's going to say go to 4. So I go to 4. Balloon-shaped body, jellyfish. Yes, it has a balloon-shaped body, so it is a jellyfish. Yes. And I'm assuming there's supposed to be a 5 that we could go to identify this little guy, but there apparently isn't one. So anyway, that's how you use the dichotomous key. You guys are going to be looking into making one of those here when we do our lab day, which is going to be pretty cool. And hopefully that'll help to clarify more on the difference between cladograms, dichotomous keys, and phylogenetic trees. But all of it, if you're feeling overwhelmed right now, just remember, all of this is to just basically make a big family tree that shows how all organisms are related to each other, which groups are more closely related, and how they are all part of the same tree of life. I hope you guys have enjoyed this, and I hope you enjoyed the practice and the extension. Have a wonderful Monday and Tuesday, and I look forward to catching back up with you guys on Wednesday. Take care of yourselves and stay safe and well.